Tēnā koutou katoa, e tino mihi ki a koutou I te wā nei. Ko te mea tuatahi, ko te karakia tīmatatanga. Nō reira, e whakawhētai ana mō tēnei rā, e whakamau mahara ki ngā taonga tuku iho, ngā maunga, ngā awa, ngā moana, ngā rākau, ngā manu, ngā koiora tuku iho. Hei tiritiri mō tātou katoa, o tātou wairua, hei hanga tangata kotahi, kia mahi tahi ai, mō te ao hou tino pairawa. Nō reira tuturu whakamaua kia tīna haumi e hui e tāhiki e. Well, kia ora koutou katoa. Welcome to the second webinar of our Biological Heritage National Science Challenge webinar series. Today's topic is state-of-the-art surveillance. Just before we get going, I just wanted to quickly acknowledge my, my fellow co-director, Daniel Patrick. He was to facilitate this webinar today, but as it turns out, he's actually having a celebratory lunch with his family. Today is the 21st birthday of his eldest son. So ngā mihi ki a rātou I tēnei wā. Alrighty, a bit of housekeeping. There's actually a webinar help guide in the handout section that you'll be able to see on your dashboard. So if you're experiencing any issues, have a look at that. In terms of the format for today, we've got 30 minutes, which I'm going to strictly adhere to. 15 minutes for a presentation, and then 15 minutes of questions and answers. So a 15-minute QA session. In terms of your questions, please submit your questions via the question section in the dashboard. And can you do us a favor and make your questions quite short and succinct? Just makes it easier for us to manage and prioritize and get to the questions. We'll do our best to answer all your questions in the Q&A session, but if we run out of time, if we get to our 30 minutes and there's still questions, we'll do our best to try and follow up after the webinar. Alrighty, so today's session, state-of-the-art surveillance. Surveillance is an essential part of protecting New Zealand's taonga species and economic assets from the damage of introduced exotic organisms. So to strengthen our biosecurity system, researchers at BioHeritage are investigating two technologies to improve the early detection of new invaders. The first of those technologies is insect soup, sampling DNA with light traps, and the second of those is spectral imaging for urban tree health. So I'd like to offer a very warm welcome to Steve Pawson, who's right in the middle of all of this, and he's going to give us a presentation and present those two technologies. So kia ora, Steve. Big welcome to you, and over to you for your kōrero. Kia ora.
Kia ora, thanks Shaun. So yeah, Steve Pawson here from the School of Forestry at the University of Canterbury. So welcoming you from a wet day in Christchurch at the moment, but talking about biosecurity surveillance today. So just to start off with, if this thing will, go forward, biosecurity risk is kind of always there. So, I mean, I guess a lot of you work in the biosecurity areas who are present at the moment. And, you know, we're importing a lot of things. And if we are going to be a trading nation, if we want to be welcoming things like these giant container ships, we have to accept that we can't possibly find every potential biological risk organism on something of that scale. You know, we're bringing in over 1 million shipping containers per year. So theoretically, 1 in a million chance events are going to happen on an annual basis. So with a backdrop of the fact that biosecurity risk is always present, we then know that there's going to be some risks that we know about. So these are some of the poster childs. I won't tell you what they are. It'd be interesting if people put in the chat if they know what they are, the left, the middle, and the right. But these are some of the things that, you know, are very high profile. Biosecurity New Zealand is very aware of them. We do risk assessment for them. We, in some cases, have specific surveillance operations for these particular things. But we also have to be very cognizant that although we have these known risks that we put a lot of effort into, there is a lot of unknown risks out there around the world. In a recent paper published by Rebecca Turner from Shaun, and if you haven't seen this paper published earlier this year in Ecological Applications, I would encourage you to take a look at it. Rebecca did some work looking at interceptions at the border across a range of countries. You can see them there in the table. and just highlighted there in New Zealand. So over a nearly 20-year period, about 70,000 interceptions were looked at, and of those we could only identify roughly about half of those to an actual particular species. So we can automatically see here that with stuff that we know that's hitting our border, we have a diagnostic problem. You know, we can't identify everything to species. And that's not to say that our diagnosticians aren't doing a fantastic job. They absolutely are. It's just that often the things that we do find, it might be a life stage that we can't identify. Maybe there isn't a tool out there that can help with that. But it's important to realize that we have this high diversity of things that are coming. So of those things that we can identify to species, the important thing to realize is that a lot of them only had one interception. So of those things that we can identify to a species level, almost half of them were only ever found once. So that's telling us that there are a lot of novel things that are hitting our shores. And that's something that's consistent globally, that there are lots of things that we just don't really know about that are potentially turning up at our borders. And given that we have all of these unknown risks, obviously what we want to do, if something's going to be a bad pest that's going to have a big impact, we want to find it early. Early detection is absolutely crucial if we want to support our eradication operations such that we can get rid of it before we get stuck in the trap of long-term pest management. So if we want to find things early, that's really hard. So another paper recently published in Australia by Angus Carnegie and Helen Narang showed that over a period from the mid-90s to about 2017, of the 34 incursions of forestry pests, most were found too late to do anything about. So, you know, Australia is a bit different to us. They have a different trade profile. They're also a much larger country. But even on a New Zealand basis, if we want to find things early, we have to do surveillance at a large scale. So if you're doing surveillance at a large scale, you've got a few problems that come on board. One is just the operational capacity and cost of doing that sampling over large areas. And again, if you're doing large scale sampling, you're going to have issues with the diagnostic side of it in terms of capacity and availability and resources for doing that. So as Shaun mentioned earlier, through what we're doing in theme 4, state-of-the-art surveillance, is we're looking at what other technologies can we use to help find things earlier. And we've taken the approach of looking at 2 different types of technologies. One that allows us to detect the signs of a particular, say, pest organism. We're wanting to try and find evidence of that specific organism. And that's where we're looking to environmental DNA, or it could also be termed high-throughput sequencing with our insect soups, and I'll talk about that in a minute. And so the aim of the environmental DNA is to be able to detect the presence of the actual organism. But also we're looking at technologies for being able to assess symptoms that there might be an incursion of something that is causing a plant health issue. And so in both of these cases, using the imaging to look for symptoms of plant health decline or looking for signs of the DNA of an organism, what we're looking for there is detections that can then inform further field inspection by inspectors. So for example, Biosecurity New Zealand already runs some large-scale surveys like the high-risk site surveillance program, and they have set transits that they do that are based on where risk is likely to be. But those transits can only cover certain parts of the country. So if we can cover bigger areas, we can then inform where inspection effort might be to supplement those existing inspection protocols. So anyway, we'll go through now the 2 technologies. So for those that are not familiar with it, this is a sort of an overview diagram that's quite nice from Curtin University that shows how you might go about doing an eDNA workflow. So it can work in many different environments. You can be looking at everything from things in the soils to freshwater, marine, or in our case, we're looking at a terrestrial system where we're wanting to aggregate our DNA. So you decide effectively for eDNA what kind of environment are you working in, then you need to have some way of collecting those samples and extracting your DNA. And then once you have your DNA, you amplify your target gene regions. We're looking at CO1 as a gene region, which is very good for resolving species level differences of insects. And then once you've got your DNA barcode, you sequence that, and then the crux of the matter is you have to then compare your sequences with your reference library of known sequences to get your species identifications. So in our case, as Shaun mentioned, we're looking at insect soups. Some work that I've been doing previously with Carl Wardhor, pictured here. We looked at a whole range of different types of traps that could be used in a port environment at a seaport, finding that UV light traps are clearly those that will suck the most species in. Poor Carl, to give you an idea of a diagnostics impediment to this, to do this particular graph there, Carl sorted through 250,000 invertebrates, of which he then took the beetles, 45,000 beetles roughly, and sorted those to these morpho species. And the light traps were the most effective, and indeed caught about 84% of all species there. So hence why we're trying, as our proof of concept in this area, to look at these kind of UV light traps for invertebrate pests. And so then obviously we go through the laboratory stuff of sequencing your DNA, extracting and sequencing DNA, and then the interesting part is obviously matching that up with reference databases. So as a proof of concept for this, our approach is first, we are going to go out and look in the international sequence databases and say, how many sequences for the approximately 25,000 pest species that are on MPI's official press register can we get a CO1 sequence for? And that will be our first cut of matching. We're not going to go and fill in a reference sequence for all of those 25,000, but as a proof of concept, given that those things that are on that list are pests, we're confident we will find a reasonable proportion of those for our first matching. Also from the previous work that I just showed there of Carl's, we have a bit of knowledge around what species we expect to see on that port, and we'll be sequencing some of those that we can't get matches for in the existing gene databases, so that we can add them to our reference suite. And after that, we'll do some broad matching with like the entire global databases to see what probability for things that we can't match, can we match them at a family level or a genus level or something. Now, one of the big advantages of using this kind of DNA approach, be it eDNA, high-throughput sequencing, is once you have your sequences from your sample, you can go back and reanalyze that at any point in the future. So as your reference library is getting bigger and bigger and more comprehensive, you can throw your samples back at that reference library to see if you can fill in any of the blanks. So the team that are working on this particular project is largely across a number of the Crown Research Institutes. So Simon Bullman from Plant and Food, Andrew Cridge from Scion, Manpreet Dami from Manaki Whenua, myself and Blair Robertson from the University of Canterbury. Blair is a mathematician, so he's helping us out with some sampling design stuff. And Shaun Wilkinson from Wilder Lab. So Shaun is the director/owner of New Zealand's only commercial eDNA facility, and he's going to be helping with some aspects of workflow stuff, but also around, we'll be spinning up next year a bit of a more community engagement approach of trying to get out and engage with the public about the use of eDNA, so we can have some discussions around some of the potential social, cultural license to operate issues, which I'll come to later. So moving on from eDNA, this is a typical scene from your urban area. If someone comes around and empties a rubbish bin, it almost looks like the street I grew up on. I actually had to do a double take. This area down here, very much like where my house would be, but I grew up in, but it's not. The one thing that I find about this picture that is a bit odd is how clean that truck is. But anyway, the reason I'm putting this up here is that if we want to do something around an image-based system to look through large areas of our urban treescape for potential pest problems, there is no way we can afford to buy ourselves a bunch of cars that are going to go out and drive the streets of Auckland and Christchurch, etc., and other areas where we have a potentially higher biosecurity risk from where, say, for example, our transitional facilities or our airports and seaports are. We can't afford a transport network like that to go around every couple of weeks. So to deal with an imaging solution like this, we need to think about how can we add to existing traffic movement. So you can kind of imagine a wee camera sitting on here on top of the cab above this guy that is imaging the trees as we go by. A much more cost-effective solution. So the approach we're taking here is take a camera, a fairly fancy camera. We still don't have our camera. There's a 6-month COVID delay wait on getting some of these cameras. We need to get what's called a global shutter camera. So if anyone has a 12 to 20 megapixel global shutter camera that they're not using, I would appreciate borrowing it for a little while. All the ones at Canterbury and Waikato are in use in existing projects. And then we'll put some lenses on that so we can get the right field of view that we want to get. We'll put it on top eventually of something like a rubbish truck, hopefully. Please don't go talking to your rubbish truck driving colleagues. We've not broached that subject with them yet, so we'll be trialing it just on some cars with tripods, etc. And hooking it up with something like GPS units so we know where those images are that are taken. And so we'll take a whole bunch of images, and you might have an image that looks like it's got a problem with it. This is just down the road from my house. This lady, I went and had a chat with her the other night to see if I could use images of her house. She's actually cut this tree down since then. Been reliably informed by a forest pathologist that it could be cypress canker, that particular thing. So the idea is we would be collecting, largely speaking, images of urban street trees and street frontages of some of the plants around the place. And then we need to do a whole pile of stuff with those imagery, privacy blurring, a thing called slamming, which is simultaneous location and mapping, so that we can create what's called like a digital twin. So every time you go back past those objects, you can say, okay, that's that tree there. You can isolate that tree in space and you can overlay or compare images over time. So once you've got this technology stack in there to do that kind of thing, you can then start to, do fun stuff with it. And there's kind of 3 fun things that we're wanting to do with that. The first one is as simple as, can we identify all the vegetation that is in those pictures? So you might come past and see a scene like this, like on the University of Waikato campus. Can we automatically say, that's where the willow trees, that's where the flax bushes are, that's where other things are? Because we know that a lot of the pest species that are coming in, say for example, I think it's the middle photo that I showed you earlier on, that likes the tree of heaven. So if you were to find one of those organisms like they did recently, there was an article in the press about it the other day, someone in Auckland, I think, popped out of some arboricultural training gear or something or other, I can't remember what it was. That thing likes the tree of heaven, so therefore you can do a quick check around the local neighbourhood of all those specific trees to see if you can find an established population. So knowing where things are is really important around a biosecurity incursion. But also, if you know of a particular pest or disease that is associated with a tree, you can start doing targeted searching for those symptoms on those trees. So as a proof of concept, we'll probably use something like this. This is an example of Dutch elm disease in Auckland. So these elm trees, these golden elms are quite characteristic. We see them all around the urban environment, as is the damage on them from Dutch elm, so quite characteristic. So where we have known symptoms of a particular disease, we can start looking to see if we can find them. So proof of concept, obviously, we'd do that with diseases like Dutch elm that are already present in New Zealand. But if we can prove that we can do that, we can start developing algorithms or classifiers for matching these known threats for things that we don't have, things like, say, Xylella, etc. But the real difficult thing, as I said earlier on, we have all these unknown threats. There are so many unknown things potentially coming over the border that we won't know what the symptoms look like. So how do we get an early warning system for those? And this was the real difficult thing that the team spent quite a bit of time thinking about how to do. So first of all, we know where all the vegetation is. So that's an oak tree. I'm not actually 100% sure that's an oak tree. I haven't been there to actually have a look at it, but let's just say it could be an oak tree. Okay, but the problem is, if you want to compare images, say every fortnight or every month of an oak tree, as we all know with oaks, they're deciduous. So during a year, any individual oak tree is going to look quite different to the computer over time during that year. So we really tried hard to think, how can we potentially identify what trees might be looking a bit sick compared to others? And so what we're going to attempt to do is to say that, okay, if you know where all the oak trees are in Christchurch, say for example, Christchurch City Council, we just literally got their tree, street tree and park database the other day, and I think there's about 3,000 Quercus ruber, so red oaks, pendulum oaks. And so if we think it could be a normal distribution, I don't know yet because we haven't gone out and imaged them all, but from an image-based classifier point of view, the bulk of those oak trees are going to look somewhat similar to each other. And then there's going to be some that are going to be way out to the sides of, do they really match with what should be an oak? And so we're hoping that those things that kind of look a bit odd might be ones that are suffering a bit from sickness, etc. And so that could be a way of informing which ones perhaps you should go and take a closer look at. And ideally that could be done digitally initially, so you've got those initial photos, but we're aware that we have to be careful with privacy. And one route to avoid privacy issues in this is that we don't actually keep any images at all. They're pre-processed on the camera, on the fly, on the rubbish truck, and the only thing that comes back is some of the classifier metrics, so you never actually keep the image. But clearly if you do that, you lose a lot of information that you could use later on. So for example, here, if you identified a particular tree that looked abnormal, you could quickly pick up an image and go, oh, it looks abnormal this time because Tree Tech have come and cut half of it down or something. Okay, let's not bother going to send someone to look for that one. So that's a really unknown area for us. We don't know how it's going to work out, but hopefully we will be able to look for more general aspects of changes in tree health. So the imaging team that's around that, I'm sort of chief cook and bottle washer there. Barbara Petrova is a data scientist, machine learning person, species classification. Richard Green is a computer vision person, as is Yi Chao and Melanie Ub and Mike Duke from Waikato. They're into sort of robotics, machine vision, etc. So this is largely a collaboration between the University of Canterbury and the University of Waikato, an opportunity also for capability building, and that we'll be supporting 4 master's students. So there's some details of those master's student projects on the BioHeritage website, and we still have some of those available. So if you know if people are interested in doing a master's in aspects of computer vision, machine learning, then we've got some opportunities for you. So as with everything really in the BioHeritage Challenge, it's mission-led. We're wanting to make a difference, we're wanting to make an impact, and so as part of doing our research, we are preparing for implementation. We're sort of wanting to develop proof of concept workflows that could be picked up by those responsible for doing biosecurity surveillance. So our focus there is largely on central government, particularly Biosecurity New Zealand, but also the tools obviously transferable into other areas like biodiversity surveying, etc. So we are also keeping in touch and talking with the likes of the Department of Conservation, etc. We're also incorporating into our research doing a business case. And so, you know, there's probably some people on the call that are here from MPI or DOC or others, but the way I view it is that our government partners are going to be interested in probably 3 key areas. One is, does the technology work? Can this find things? The other is, how much will it cost? Is it cost effective to do this? Hence why we're doing the business case. And the third is, you know, do we have a social cultural license to operate in this space? And we're aware of those issues and starting to spin up some pieces of work around that. From the DNA perspective, obviously there's some interest there from, well, who should have access to sequence data and custodianship of that data. And then from the imaging side, clearly it's around the privacy. And there are very good technologies there that, for example, Google Street View use. But then Google doesn't have the same sort of obligations to New Zealand citizens as would a New Zealand government initiative to be imaging urban treescapes. Some of these things are areas where there's already a lot of adoption or deeper research going on internationally, particularly with the eDNA space. And most recently, we've been talking with Australia and the MPI equivalent in Australia, as the Australian government has just announced a $7 million investment in developing eDNA technologies. So members of our team have been speaking both with researchers and participating in conversations with government in Australia around what they are doing in the space of getting towards a more implementation, sort of operationalization of these particular tools. And that's where I'll leave it, Shaun.
Kia ora, thanks for that, Steve. Very interesting stuff. Just a note for the attendees, if you want to add any questions into the questions part of in your dashboard, please throw those in. I know we've only really got a few minutes, so I'm just going to kick things off and ask a question of Steve. Steve, so on a global scale, are there examples of the technologies that you've talked about that are currently actually being implemented in an operational sense that you could tell us about?
Yeah, so there's a little bit. So from the imaging technology, there's a few people who are starting to look at, you know, vehicle-mounted cameras, particularly for that first stage of what we're looking at. Can you identify particular plant species? And people are looking at particularly from finding weed species on roadsides, etc. So there's a bit of experience there that we can tap into. From the eDNA side, yes, that's starting to become a bigger feature, and definitely these collaborations with Australia, as I say, this $7 million investment includes both research, but also a whole pile of stuff around developing other things like protocols and guidelines for how you should develop an eDNA-based test for biosecurity, how you should validate the outputs from those, how you should certify laboratories for doing those kinds of things. So they're doing a lot of the work already on how might you implement something that we can piggyback off to learn from their experiences, which I think will be very useful for MPI, and they are actively engaged in those conversations, which is great.
And just a really quick follow-up question, I guess in 1 minute or less, but what are the parts of the research that excite you the most?
Well, it's all exciting. I mean,
It's all exciting. It's all exciting.
In with it, but I think to me that the biggest unknown and most exciting little bit is that bit on the imaging around where we're looking to just try and find signs of general ill health in a tree, because, you know, everyone knows who's involved in biosecurity that, you know, as soon as you put it on a list, it puts it up in those big flagpoles of known pests. But we also know that by and large, it's things that are not on the list that come and bite us in the end. So having an ability to have a more general thing that's just looking and able to pick up unknown threats early so we can get a handle on them is really exciting. So I have no idea if it's going to work, which is even more exciting. So more fun.
Cool. I just I see we've come to the end of our time. If people need to jump out, please do that now. We'll just maybe go over just a few minutes. I just want to there's a comment there from Brendan Gould that I think is just worth running over, and he says, great overview, thanks Steve. One of the biggest challenges with the technology is the ability to manage, retain and analyze the data, which generally requires significant investment in IT and information management systems. Are you looking at how to support this tech with affordable technology? So it's an affordability question.
Yep. So yes, indeed, sampling is extensive. You know, we have chosen on the imaging side of it to go down the route of using a simple RGB camera. So that's effectively the camera you have in your cell phone, red, green, blue, standard visual camera. People have probably heard of multispectral, hyperspectral cameras. Yes, a hyperspectral camera may allow you to identify disease slightly earlier, symptoms earlier, but the cost of buying those cameras, calibrating those cameras and developing the algorithms with them is just so much more complex. So hence we've gone for proof of concept starting at the lower tech end. Even so, you know, 10 hours of recording of that, you know, we're going to be up to, you know, 15 terabytes worth of data. So, you know, you're quickly going to get up into large amounts of storage. But as I say, you don't necessarily need to keep all of those images. From the eDNA side of it, we recognize that there are some of these supporting infrastructure things, as you've been talking about there, that are beyond BioHeritage, and are things that I think through this collaboration between the Australian government and New Zealand government, we'll get an idea around some of those. And in particular, those are, the big one is having a national reference collection of sequence information. So Australia is just in the process, or rather CSIRO in Australia are launching a national biodiversity sequence library, I think it is, I can't remember, and they have a goal over 10 years to sequence all of the known species in Australia from their collections. They estimate it'll cost them about $40 million, and they're currently entering into partnerships with various government providers like their MPI equivalent over there to rapidly build that collection from New Zealand specimens, and they're already several tens of thousands species into it. So that is the big crux of getting an eDNA library. But of course, if other countries are doing that as well, we obviously share a number of species, so we can benefit from their work as well. But that is probably the big underlying infrastructure that New Zealand needs to think about, is how do we develop that library for our uses here in New Zealand.
Kia ora, thanks for that, Steve. Alrighty, I'm aware that we've gone over time a little. I just want to take just a little bit of extra time to say thanks to you, Steve, for your coverage of this awesome topic. Thanks to all the attendees for attending and taking part here. I want to wish you all, on behalf of the Biological Heritage and on behalf of Steve and myself, actually, I wish you all the very best for the coming holiday break. Make sure you have a well-earned break. I think we say that for all of us, have really earned a break over this year that's just gone. I'll finish us off with a karakia. Nō reira, whakamaua te kōrero, whakamaua te wānanga, whakamaua kia rangi nui e tu iho nei, whakamaua kia papa tuanuku e tākoto ake nei, whakatīnā ki te tumu o te māramatanga. Nō reira, kia tīnā haumi e, hui e, tāi ki e. Kia ora koutou, kia pai o koutou wā whakatā. Ka kite.